Hey everyone, and welcome back to The Explainer. So today, we're unlocking one of the absolute coolest subjects in modern science. We're talking about biotechnology. Now, right off the bat, I want you to completely change how you look at a living cell, okay? Don't just picture it as some microscopic blob of jelly. Instead, I want you to think of it as a highly complex, unbelievably efficient manufacturing plant. And just like any massive factory out there, it runs on a very strict set of instructions, basically a blueprint. In this explainer, we're going to see exactly how scientists have figured out how to read, cut, paste, and literally rewrite that biological blueprint to make cells produce amazing things, from life-saving medicines all the way to pest-resistant crops. So let's jump right in.
To kick things off, we kind of have to ask ourselves, what actually counts as biotechnology? I mean, it's a huge umbrella term. Technically speaking, humanity has been using live organisms to make useful stuff for thousands of years. Think about making curd or baking bread or fermenting wine. Those are all micro-mediated processes. So in a very broad sense, your grandmother making curd in the kitchen, she's actually practicing a traditional form of biotech. But, today when we use that word, we're usually talking about something much more restricted. When we say modern biotech, we mean using genetically modified organisms on a massive scale, or using highly advanced lab techniques. So things like in vitro fertilization to create a test tube baby, synthesizing brand new genes, developing a DNA vaccine, or even fixing a defective gene. All of that falls squarely under the modern biotechnology umbrella.
Because there's this split between the traditional view and the modern molecular view, the European Federation of Biotechnology, the EFB, stepped in. They created a comprehensive definition that actually bridges both ideas. Now, listen up, because if you're a student prepping for competitive exams like NEET or CUET, you're going to want to pay super close attention here. This exact phrasing is highly, highly tested. The EFB defines biotechnology as, quote, the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. Definitely memorize that one. It just perfectly captures the whole vibe of taking the raw biology of an organism, mixing it with pure natural science, and creating something incredibly useful for us.
Here's our roadmap for today. First, we'll open up the Biotech Recipe Book. Second, we'll dig into the Genetic Engineering Toolkit. Third, we'll meet the delivery vectors. Fourth, we'll go through the steps of gene cloning. Fifth, we'll look at PCR, which are basically biological photocopiers. And finally, we'll wrap up with biotech in our world.
All right, let's start with Section 1, the Biotech Recipe Book and its core principles.
So the whole birth of modern biotechnology was basically driven by two core techniques. If we go back to thinking of ourselves as a factory kitchen, well, we need to know how to rewrite the actual recipes, and we need to make absolutely sure our kitchen stays spotless. Let's break those down. First up is genetic engineering. This involves the actual techniques used to alter the chemistry of genetic material, so the DNA and RNA. This is where we write a brand new recipe and introduce it into the host organism, which fundamentally changes its phenotype. Then, the second part is bioprocess engineering. Look, you can have the absolute best, newly engineered cell in the world, but if your manufacturing vat gets contaminated by some random wild bacteria, your product is completely ruined. So bioprocess engineering is all about maintaining a sterile, microbial-free environment. That way, only our specific desired microbe or eukaryotic cell grows in those massive quantities. And that's absolutely crucial for manufacturing biotech products like antibiotics, vaccines, and enzymes.
Moving right along to Section 2, the Genetic Engineering Toolkit and our molecular tools.
If we're going to go in and alter the DNA recipe, we need some very specific molecular tools to get the job done. The very first tool we reach for are our molecular scissors, which are formally called restriction endonucleases. These are just amazing little enzymes. They literally scan a DNA molecule and snip it at very specific, predictable locations. But here's the problem. After we use our molecular scissors, we're left with this huge jumble of DNA fragments of all different sizes. So how do we isolate the exact one we want? Well, we use a super clever technique called agarose gel electrophoresis. See, because DNA fragments are negatively charged, we can put them in an electric field and force them to move toward a positive anode right through an agarose gel matrix. It's kind of like an obstacle course. The smaller fragments are nimble, so they slip through the gel way faster and move further down, while the big, bulky ones get stuck near the top. As the digested DNA separates beautifully by size across the gel, it lets us pluck out our specific gene of interest.
Okay, now that we've successfully cut our desired gene out, we have to paste it into a new piece of DNA, and that's where our molecular glue comes in. We use an enzyme called DNA ligase, which perfectly joins those cut ends together. When we use our ligase glue to paste our gene into a new DNA molecule, we create something called recombinant DNA. Guys, this is a massive exam term, so highlight this in your notes. Recombinant DNA is essentially a new combination of circular, autonomously replicating DNA created in vitro, which just means it's made in a test tube outside of a living organism. Fun fact, the very first artificial recombinant DNA was constructed by two scientists, Herbert Boyer and Stanley Cohen. They actually managed to link a gene that coded for antibiotic resistance straight into a native plasmid of Salmonella typhimurium. Pretty groundbreaking stuff.
All right, Section 3. Meet the delivery vectors, or as I like to call them, the delivery trucks.
So here is the catch. That fancy recombinant DNA we just made, it's completely useless if it's just sitting around in a test tube. It absolutely has to get inside a living host cell so it can multiply and actually start manufacturing our product. For that journey, we need our delivery trucks. In biology, plasmids and bacteriophages are incredibly useful delivery vehicles because they have this awesome ability to replicate inside bacterial cells totally independently of the chromosomal DNA control. A really perfect example of a biological delivery truck is the PBR322 cloning vector. Seriously, if you're a student, you should probably have this diagram etched right into your brain. This specific plasmid has been highly engineered to help us easily link our foreign DNA and then figure out which cells actually swallowed up the new blueprint. If you look at the different labeled regions on this circular DNA, basically these are specific docking sites where our restriction enzymes can make a cut, opening the plasmid up so we can sneak our alien DNA right inside.
So the really crucial point here is understanding the specific features packed onto that PBR322 vector. The two biggest ones you need to know are the ORI and the selectable markers. ORI stands for origin of replication. This is quite literally the sequence where replication starts. If you want your alien DNA to multiply, there is no way around it. It absolutely must be linked to an ORI sequence. Plus, the ORI controls the copy number. So if you want a ton of your product, you got to clone it into a vector whose origin supports a high copy number. Make sense, right? Secondly, we have our selectable markers. These are things like the antibiotic resistance genes, which are labeled AMP-R and TET-R. Think of these as biological name tags. They help us easily identify and select the specific host cells that successfully took in our recombinant DNA, while letting us eliminate all the non-recombinants that didn't.
Which brings us to Section 4, Steps of Gene Cloning, the actual manufacturing process.
Okay, we've got our tools and we've got our delivery trucks. Now let's actually put this whole factory manufacturing process together. If you're ever sitting in an exam and asked to outline how to genetically modify an organism, there are 3 fundamental steps you absolutely must know. Step 1, identification of DNA with desirable genes. First things first, we have to find the specific blueprint for the product we want, say the gene for human insulin. Step 2, introduction of the identified DNA into the host. This is where we grab our restriction scissors, our ligase glue, and our plasmid delivery trucks to physically get that gene into a host factory, like an E. coli bacterium. And finally, Step 3, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. We need that E. coli to survive, replicate, and pass our brand new blueprint down to millions and millions of new bacteria. That way we build up a massive workforce that's fully dedicated to producing our product.
Next up is Section 5, PCR, or what we like to call the biological photocopiers.
Now, sometimes we run into a little problem. Often, the sample of DNA we extract that contains our desirable gene is just way too small to actually work with. But don't worry, that brings us to one of the most revolutionary, game-changing tools in all of biotech, PCR, which is basically our biological photocopier. PCR stands for polymerase chain reaction. Let's take a really close look at how this cycle works. Basically, in this reaction, multiple copies of our gene of interest are synthesized in vitro. We use 2 sets of primers, which are just these small, chemically synthesized oligonucleotides, and an enzyme called DNA polymerase. Through repeated, rapid cycles of heating and cooling, the enzyme extends the primers using nucleotides provided in the mix, using the original genomic DNA as a template. Get this, if we repeat this process just 30 times, we can amplify a single, tiny segment of DNA roughly 1 billion times. It is just a ridiculously powerful amplification tool.
To make sure you have this totally locked in for your exams, let's explicitly run through the 3 steps of every single PCR cycle. Step 1 is denaturation. This is where we blast it with high heat to separate the double-stranded DNA. Step 2 is primer annealing. Here, the temperature is carefully lowered so our little primer tags can actually attach to the target sequence. And step 3 is extension of primers. This is where the DNA polymerase enzyme races down the strand, building the brand new copy. So again, denaturation, annealing, extension. Seriously, remember that sequence.
Finally, let's head to Section 6, Biotech in Our World and the Final Products.
So let's see how all of this microscopic science actually builds into the real world. Once our tiny microbial factories are happily producing the exact protein or organic compound we want, we use these massive bioreactors and incredibly strict downstream processing to purify the product on an industrial scale. But what exactly are we making out there? Well, the applications of biotechnology are just vast, and they deeply impact our daily lives. On the medical front, we're churning out massive quantities of therapeutics, like biopharmaceuticals, and developing hyper-advanced diagnostics. Over in agriculture, we are creating genetically modified crops that are specifically designed to yield more food and withstand super harsh conditions. Environmentally, we're using engineered microbes for bioremediation and waste treatment to clean up nasty stuff like oil spills and toxic waste, and we're even utilizing them to generate sustainable energy. It's truly everywhere.
Just take a look at this striking visual. What you're seeing is genetically modified cotton, specifically a type known as Bt cotton. By taking a specific, naturally occurring toxin gene from a bacterium and cloning it straight into the cotton plant's DNA blueprint, literally using the exact same techniques we just talked about today, scientists engineered a plant that produces its own internal pest resistance. I mean, compare a healthy, fluffy cotton ball to an absolutely destroyed, pest-ridden one. This isn't some futuristic science fiction, folks. This is applied biotechnology, right here, right now, securing our agricultural yields against devastating insect attacks without having to rely on dumping heavy chemical pesticides everywhere.
We have truly managed to transform tiny, completely invisible cells into the most sophisticated, high-tech manufacturing factories on Earth. From using molecular scissors to paste life-saving genes into delivery plasmids, to literally photocopying DNA billions of times in a few hours, the toolkit of biotechnology is just astonishingly powerful. We've engineered cells to make vital medicines, clean our toxic waste, and protect our global food supply. But here's the crazy part. This technology is only accelerating. So the real question I want to leave you with today is this. What organism will we reprogram next to solve humanity's absolute greatest challenges? Keep studying, keep asking the big questions, and I'll catch you in the next explainer.